Hello, everyone. My name is Cornelius Christopher Nadio. I'm from Biotechnology Cohort 2025, and today I will be giving a presentation about the use of actinobacteria in bioremediation of oil waste. Now, a major problem all over the world right now is oil waste. Now, oil waste can be originated from either oil effluents from factories across the world or oil spillages. Now, these oil wastes can actually cause a multitude of problems, such as damage to sea life for creatures such as fish, crabs, and other aquatic creatures, as well as the pollution of multiple habitats such as forests, coral reefs, and other environments. Now, the pollution of habitats can directly lead to the next problem, which is animals and humans can actually get sick due to these harmful chemicals. Now, these harmful chemicals from the oil waste can seep into the ground where the plants grow and are then consumed by animals, to which then they digest the harmful chemicals that were absorbed by the plants, that causing more animals or humans to get sick from consuming those animals. Harmful chemicals from oil waste can also seep into water supplies, causing more humans and animals to get sick. Now, these dangerous compounds include amines, heavy metals, benzene compounds, and other dangerous compounds. One of which is petroleum hydrocarbons, which is very harmful and can cause severe health issues. So petroleum hydrocarbons can be measured in TPH, or otherwise known as total petroleum hydrocarbons. Now, despite the multiple efforts for oil waste removal across the world, unfortunately, the current conventional methods that are being utilized are ineffective. Methods such as gravity separation and flotation are unfortunately inefficient and high in operational costs, which makes them not suitable for these oil waste removals. Now, this is where actinobacteria comes in. Now, actinobacteria is a filamentous gram-positive bacteria that is mostly aerobic, but can also be facultative anaerobic or strictly anaerobic. Within their DNA, they contain 70% GC content and can also survive in extreme conditions such as dry environments or nutrient starvation. They can also produce biosurfactants that increase the contaminant bioavailability and help facilitate the biodegradation process. However, there are still research gaps relating to the use of actinobacteria in oil waste removal processes. These include the insufficient research and unexplored ideas regarding the topic, which ask whether using actinobacteria for oil waste removal is comparatively better compared to other conventional methods, as well as the lack of real world testing for the methods that use actinobacteria, which brings the question, how effective is actinobacteria in methods for oil waste removal compared to other conventional methods, and whether actinobacteria is viable in the real world environment. Now, these questions will be answered in this presentation. For the methodology that was used for the research of this presentation, the sources were obtained from both Google Scholar and Consensus. Now, the criteria for the reference that were used include the year range being from 2016 to 2026, so that the data from the articles and journals used are up to date. Another criteria would be that these articles and journals that are used focus on the topic of oil waste, environmental issues, bioremediation, as well as actinobacteria. And finally, one of the other criteria for the references is that the journals and articles use English as their primary language. For the two tests that were conducted, The first test was the comparison of degradation of petroleum hydrocarbons, or TPH, between bioremediation methods and conventional methods. Now, the methods that were chosen for for the methods that use actinobacteria, or otherwise known as bioremediation methods, include biostimulation and bioslurry. As for the conventional methods that were used for comparison, include ultrasonification, flotation, microwave frequency heating, and thermal desorption. As shown in the bar graph displayed here, the methods using actinobacteria were either equally or more effective than the other conventional methods that were used. However, the advantage of the methods that use actinobacteria were that they were more cost effective and required less energy, or and especially for bioslurry, the method is time efficient compared to the other methods. The conventional methods that were not as effective were ultrasonification and flotation. While they were commonly used, as displayed in the bar graph, they were not equally effective compared to biostimulation and bioslurry. Now, for the methods that were more effective, such as microwave frequency heating and thermal desorption, the microwave frequency heating method utilized a lab control environment, so could not be used in the real world environment. And for the thermal desorption, it required high energy, despite it being the most effective compared to all other methods that were used here. The second comparison that was done was the comparison of bioremediation using actinobacteria in different environments. The two different environments were the lab controlled environment and the real world environment. This comparison was done to see whether actinobacteria was actually, the methods that use actinobacteria were actually effective in the real world environment. The first factor was the TPH degradation or removal. Now, in that factor, the real world test actually achieved a higher result compared to the lab controlled environment. As for the other factor, which was the oil removal test, the lab control environment actually instead achieved a higher result compared to the real world environment. Though the most important highlight from this testing is that both tests actually showed that the usage of actinobacteria in bioremediation process is still effective in the large scale real world environment, just as in the lab control environment. So, in conclusion, the use of actinobacteria for bioremediation for oil waste removal is a viable and effective option, which is also cost effective, can be time efficient, and requires much less energy compared to other conventional methods. The use of actinobacteria in bioremediation process is also still effective in the large scale environment, which is the real world environment, making it a viable and suitable option for oil waste removal across the world. However, there are still certain things that can be covered in future discussions, such as what is the most effective type of actinobacteria for oil waste removal or management, which method is suited for each species of actinobacteria for it to be effective in oil waste removal, and what combinations of bioremediation methods that use actinobacteria can be used to yield the most effective result for oil waste removal. For the AI declaration, the AI tool consensus was used for researching the relevant articles and papers that were related to the topic being discussed in this review, this presentation, as well as Google Gemini was also used for the clarification of data and information regarding the research of this topic. And finally, MyBib was also used for the citation of the reference used in this presentation here. The references can be found within this QR link, this QR code, and that will be all from me. Thank you for your attendance.